all, and maybe JP, because Jenny Delaney, who's now working with me, guilted him into it. But really, I appreciate you all being here, and thanks for the invitation. This is great. What a cool place. I've enjoyed getting to see your digs and learn about what a lot of you are doing. So yeah, so thanks. I wanted to start by sharing a little bit more about myself and kind of explain the title a bit. But you know, as Katie mentioned, I ended up working with Jim Childress along with Brad, and Brad can tell you all about life in the Childress lab, but it's really an animal physiology lab. And I went there with the intention of studying the physiological and biochemical adaptations of animals to life at hydrothermal vents. And they're incredibly cool organisms. And during my PhD, quickly figured out that a lot of what I was doing was microbial physiology as well, studying their symbionts. And it's a line of research that we kind of continue today, along with Katie, who were funded together via NSF to work on these things. I won't be talking to you much about that because you all have Katie here. And I know she just gave a talk recently on this, and you'll probably be hearing more in the future about her exciting work and the cool discoveries she and Juliana and others in the lab are making. But when I think about adaptation to life in the deep sea, what really comes to mind is the sort of physiological and biochemical mechanisms by which organisms flourish in their particular environments. When I started working with Ed, it became apparent that microbial physiology and comparative animal physiology were really two different schools of thought. They really take different approaches to address the questions that are at hand. And it was really fascinating to learn about how we might apply the sort of molecular-oriented approaches to microbial ecology and ecophysiology, right, to some of the questions we have about symbiosis. And conversely, about how microbial physiologists could learn some of the techniques and concepts and ideas that comparative animal physiologists have developed, frankly, since the days of Galileo, who himself was a physiologist. So a lot of what happens in my lab is it really cross-cuts domains and focuses on this question of what is the nature and extent of the relationship between an organism and its environment, right? And it's sort of inextricable. And I think if you really want to understand organismal ecology, evolution, you have to look at their physiology. And that has been a theme running through my lab. I'm a bit of a gearhead as well, and so we've built a number of different tools that we hope advances our ability to do that kind of work. So today I wanted to just give you a brief introduction to the deep sea for those of you who may not think about the deep sea as frequently and obsessively as some of us. And I'm going to tell you about 3 different vignettes, and the first 2 are going to be a little longer, the third will be shorter. I'm going to tell you about work we've been doing on iron oxidizing bacteria, and trying to better understand how they make a living in this very oxygen-replete world. And I'll tell you a little bit about some relatively recent work on whales and their microbiomes, and then I'll share a bit about the kind of technology developments we do. If I start running out of time, I may skip through a few slides, so I hope you'll forgive me for doing that, but we'll happily answer questions afterwards. But this is one of my favorite quotes from Arthur C. Clarke, right? We really shouldn't call our planet Earth, we should call it ocean. You know, and the deep sea in particular, though it be out of sight and out of mind, it's a major part of our planet's habitable space. And by deep sea, we're talking about the ocean that's 1,000 meters or so below sea level, a kilometer below sea level. The average depth, as many of you probably know already, is about 4 kilometers. To refresh your memory on this, at its deepest point, you're looking at a pressure of about 8 tons per square inch. This is about the equivalent of any one of you, if I were to take your total body surface area, would be about the equivalent of balancing 50 jumbo jets worth of pressure uniformly distributed across your body. So pressures are immense. The longest mountain range in the world, mid-ocean ridge system, is in the deep sea, some 65,000 kilometers or so in length. 90% of our planet's volcanic activity occurs in the ocean. And I imagine there's a few trivia buffs out here. Anybody want to take a guess at how much, let's call it dissolved or suspended gold there is in ocean water? How much is there, if we could get it all out, how much gold could we give each person on Earth? Anyone want to take a guess? A gram? A kilogram? A pound? Okay. I heard one pound. What else we got? 100 pounds? 1,000 pounds? What do you guys think? It's about 9 pounds, right? So 7.8 billion people or so on Earth, there's enough gold that you could give each of them just under 5 kilograms or about 9 pounds of gold. Right, so just and the intent there is to provoke any of you who are entrepreneurial in spirit to give this a shot and remember me, right? If you just don't forget the little people, but also just I think this is a great way of illustrating just how expansive the ocean is, right? I mean, we all know there isn't so much gold in seawater that you're going to go out and get it out, right? You're talking like trace amounts, but if that it gives you a sense of the scale of the ocean. The deep sea itself is 80% of our biosphere by volume. So everything else you think of, every other habitat, including the upper kilometer of ocean, all of that is 20% of our planet's living space. And the rest of it is this cold, deep, dark ocean, right? That many of us think is exotic and extreme and weird, but I would argue if it's 80%, maybe these are the normal ones on our planet, right? Something to think about. And of course, it harbors a lot of microbes. This is near and dear to my heart. There's about 10 to the 27th microbes in the ocean. I'm going to leave viruses alone. Maya can tell you all about that. But they weigh about 10 to the 14th or 10 to the 15th grams. What blew my mind is I asked, well, how long is 10 to the 27th microbes? It's 10 to the 21st meters. That's 105,000 light years. That, my friends, is our galaxy. I thought that this was going to be like here to Uranus or, I don't know, Saturn or, I don't know, maybe a nearest star. But this kind of, again, gives us a sense of scale and immensity. And so though they may be small, they're mighty, right? There's so many of them that we have to constantly remind ourselves about the role that they are playing in running our planet, and frankly, how little we know about them. So why do we know so little about microbes? Well, there's really two answers to that. One, in many ways, we're learning a lot about microbes. The sequencing kind of revolution, if you will, was a watershed for microbiology. In that we could finally peer into the lives of these organisms that have eluded us for so long. Right now, I think there's over 30,000 microbial genomes available to you. 30,000 genomes, right? That's amazing. And this number, by the way, just a few years ago was 12,000, and a few years before that was 8,000, and then 6, and then 1, and then a handful, right? But despite that, the vast majority of microbes have eluded cultivation. So what that means is we can't grow them. And that is a major problem, because how do you really understand an organism that you can't really poke or prod? You know, it's one thing to study a panda bear. It's quite another to try and go out and look at one microbe in a soup of 1 million microbes in a teaspoon of seawater. And that's the big challenge. And really measuring microbial metabolic activity has been a slow process. I will shamelessly plug the need for more and more metabolic rate measurements, because at the end of the day, that is something that we need to critically understand, especially in our changing world, whether it's a microbe or an animal. Did I do well, Brad? Thank you. And for microorganisms, we're often collecting these data using discrete samples, and it's a slow process. And at the same time, our understanding of microbial ecology and evolution through this omics lens is growing by leaps and bounds. So Ed DeLong coined this phrase in a review article that I love. It's an impedance mismatch, and it's a riff on an electrical engineering term, meaning things are happening at two totally different rates, that it's hard to kind of line them up. And I think that is one of our grand challenges in microbiology right now, is how do we get from microbial identity, activity, to biogeochemical cycles in a meaningful way that allows us to better understand these microbes on our planet. And I would argue that many of us are now engaged in so-called omics-informed studies, where we're using the various omics technologies to get a snapshot of an organism or microorganism, and to say, okay, these are the kinds of genes or proteins that are being expressed. That's great, but don't kid yourself, that is not a rate. Even changes in the abundance of proteins is not going to tell you anything about the rate of a particular process. So using those omic tools to guide our physiological experiments is something we try and do. And in my lab, the big focus is really allying microbial identity and function to biogeochemistry, right? And that's where we bring these tools together. So again, I'll tell you a bit about iron oxidizing bacteria in aquatic environments, tell you a bit about whales, and we'll end with this vignette about our instrument development. So first, I'll tell you about iron oxidizers. I'm terrible at coming up with titles, so I'll just kind of tell you right now, shocking news is because they do extracellular electron transfer. I thought shocking would be funny. Let's just get that out of the way because it's really not that funny. But there's a lot of interesting things about iron oxidizing bacteria that I want to share with you. And let's start with the fact that iron, of course, is the 6th most abundant element on Earth. It's something that we all have a pretty good intuitive understanding of. Iron is a key part of our steel and metal alloys. Many of you here in St. Pete are familiar with rust, which of course is iron oxide. And in our oxic world, iron oxide is like the predominant form of iron, at least in, again, where oxygen is present. And that reaction happens really fast. Oxygen loves iron, and iron is very good at interacting with oxygen. So that's something that happens briskly. And it's a key part of the stories I'm about to share with you. Despite the prevalence of those abiotic reactions, microbes still participate in iron cycling, and they play a very important role in iron cycling. I'm going to keep it simple and talk about iron oxidizers and iron reducers. Iron reducers are those organisms that breathe iron oxides. Interestingly, how many of you have heard of microbial fuel cells? A few of you? Many of you? Great. So basically, in parts of the sediment, marine sediments in particular, and other anaerobic, or rather anoxic places on Earth, where there's no oxygen present, there are often metal oxides. And microbes can use those in lieu of dissolved oxygen or dissolved nitrate or the like. And really, there's been this watershed of research in this area of extracellular electron transfer that started with the study of iron reducers. So let me explain this real briefly, and I'll go on to the next aspects here, but let's talk about iron reduction real quick. If you're a microbe living in an anoxic marine sediment and there's organic carbon and there's rust, well, that rust could be a great electron acceptor, but you have to get to it, and it's insoluble, right? It's this particle that you can sit upon or next to. So you can't really bring it into your cell. So it turns out what microbes have evolved is a capacity to move electrons from central metabolism inside the cell to the outside of the cell, onto that iron oxide. A silly analogy would be like after the holiday or in the holiday party, we're like, oh, there's not enough oxygen here, and you put your hand on a rusty plate and you breathe that, right? That's what microbes do. So they breathe these iron oxides, and that's the basis of the microbial fuel cells, which I hopefully will have time to mention later. Others are involved in iron oxidation. So they're taking reduced iron and they're oxidizing it with oxygen, right? Now remember, I mentioned that iron oxides are abundant in our biosphere because it's of our oxygenated Earth, but reduced iron, at least on the surface, is less so. And iron oxidation is largely abiotic, and it's interesting because it's autocatalytic. That means that rust begets more rust, to use a biblical, riff there. So there are these microbes called photoferrotrophs that are really cool. They don't live in the deep sea, but they live in shallower aqueous environments. We see them in estuarine runoffs, shallow lakes, tide pools, tidal flats and the like. But photoferrotrophs are cool. They use the energy from sunlight and the electrons from iron 2, and then they produce iron 3 to maintain, for the most part, autotrophy, right? So they can fix carbon like a plant does. But this is what's really interesting. These microorganisms, they need sunlight. They absolutely need it. They're photoferrotrophs, okay? But in sunlight, at the surface here, they're competing with this rapid abiotic oxidation of iron, which really stinks. And it's been a real challenge. And people have tried to figure this out for a while because, again, the end products of their activity, the iron 3 that they produce, facilitates the abiotic oxidation. It's autocatalytic. It gets even stranger. They encrust themselves as they grow in iron oxides, beginning with goethite that eventually leads to hematite or magnetite. And they sort of make it even more difficult for them to access iron 2. So let me sort of step back for a second. So if you're sitting here in this rusty pond, most of the iron 2 is going to be deeper in the sediments, where it's anoxic. So why not live down there? Well, there's no sunlight. And as soon as you come up to the surface waters, your iron 2 is largely gone because it's being abiotically oxidized. But there's sunlight. So it's a bit of a pickle, isn't it? So where do they live? Well, they live, you know, people say, oh, they live in these little fine interfaces where those two are in balance, and there's likely truth to that. But we started to ask the question, well, maybe extracellular electron transfer can help alleviate this issue. And the model here was this. If you can actually see iron reducers shuttle their electrons from central metabolism out to an iron oxide, is it possible that iron oxidizers can actually take up electrons or their equivalents from the environment? And let me explain how we tested this. We started working with a bug called Rhodopseudomonas palustris. This is one of the few genetically tractable iron oxidizers. And there's it's been pretty well studied. It can live aerobically, anaerobically, and photoautotrophically and the like. And we're just focusing on this photoautotrophic part, where they, again, need iron 2, and they need sunlight, and they need CO2, and somehow they get all of these in nature and manage to compete against the abiotic process. We know that in this particular bug, without burdening you with the details, there's a system called the PIO system, right, that's involved in this process of iron oxidation, because if you knock it out, they can't oxidize iron. What we did is we started looking at these microbes using these tools called microbial fuel cells, right, or bioelectrochemical reactors. And here was the idea. The idea was, if we do these experiments with iron 2 and iron 3, the abiotic chemistry gets so complicated so fast, we can't test our hypothesis. And our hypothesis was, they are taking up electrons from the external environment as a way of avoiding the challenges of being in two places at once. Or another way of saying it is, if they could live where there's sunlight and somehow gain access to those electrons from iron 2 without being near iron 2, maybe that works. That was a hypothesis. And the way we tested this is by using an electrode that we poised at a potential comparable to the iron 2, iron 3 couple. So we basically tricked them into thinking this electrode was iron 2. And we incubated them at the pHs that they encounter in situ and so on. And this actually allows us to deliver electrical current and measure how much current is taken up on microbes that are wild type versus those in which we've knocked out PIO, ABC, and so on and so forth. Everybody okay with that? Have I lost anyone? Okay, so it's a way of testing this question. Can they take up electricity, right, and use it? So the answer is yes. Taiwan does take up electrical current, and we found that when we look at the current densities of the wild type in the presence of sunlight, they're taking up about a microamp per centimeter squared. This is actually an awful lot for the density of the microbial culture we had, so they're actually really quite good at this. Notably, they will also do this in the dark. This is something that nobody had really ever considered before, and it's a little unclear as to what the fate of these electrons are. In the presence of sunlight, these get a little boost through photosystem up to a higher potential, so they can use it to reduce ferredoxin and do the kinds of things that autotrophs do, right, to convert CO2 to sugars. This is interesting, but let's set that aside for now. What struck our attention is that when we knocked out, sorry folks, I got these labels backwards. This is the wild type, this is PIO-ABC. What you need to take away is that when we knock out PIO-ABC, we see a 30% reduction, but it doesn't stop it. So we cannot implicate PIO-ABC as to being the only mechanism involved in this iron uptake. But what's interesting is that when you knock it out, you don't see the reduction of iron 2. I'll come back to that in just a second. I know this is a little bit confusing, but, and I'll sort of skip over this. But what's really cool is that this bug will grow autotrophically in these reactors. So, and I want to explain what's in these reactors. They are growing autotrophically on CO2, on sunlight, and electrical current in minimal media. That's pretty amazing, right? To have them be able to grow basically in the presence of just enough sort of trace elements and micronutrients and the like, but really they're deriving all of their reducing power from this electrode in the form of electron uptake. And when we do that knockout with that PIO system, we see this 30% reduction, but they're still taking up electrical current. Growth is hindered proportionally, but they're still capable of growth. Now, what this tells us, to kind of get to the punchline here, is that the PIO system is specifically involved in this process of iron oxidation. But we also know, interestingly enough, that these microbes we now know they can grow on thiosulfates. And what's really cool is all of this comes together in a kind of a really nice story, again, that tells us about what these organisms are doing. They have outer membrane constituents that are electroactive. They will actually, these are sort of tells us about the redox potentials at which the midpoint potentials of these outer membrane factors, including PIO and some others. But the punchline here for this organism goes something like this. Here's an electrode. We have the PIO complex that we know is involved in some sort of degree of electron uptake, about 30%, and they can derive this from iron. There are other factors here, potentially soluble mediators, that's sort of our current guess, that's coming up and taking electrons from the electrode, and they're being taken back up into the cell and bringing in electrons that way. That explains why we have observed this organism capable of growth not just on iron 2, but also thiosulfate, something else that's chemically reduced and can give electrons, right? So PIO does seem to be related to iron, but clearly they're taking up electrons through other means. What does all this mean? Like, who cares, right? I'm coming up here and telling you about whiz-bam, this and that. But the ecological relevance, I think, comes down to this. This is sort of how we thought about photoferrotrophy before. You've got a cell. It has to be in the photic zone. You've got light. And we assumed magically somehow that iron 2 was present up here. And this was the sticking point, folks, because in the photic zone, that iron 2 is going to disappear really quickly. That's what was nagging us. Understanding now that they're capable of extracellular electron uptake, and bear in mind, remember I told you that they produce these abiotic minerals, hematite and magnetite? Those are conductive, or at the very least semiconductive. But they will conduct electrical current, and they grow the stuff all around them, and they form these sort of long matrices, which means now, through magnetite or hematite rather, they're actually in electrical continuity with the environment below the photic zone. And that means that they have electrical access, more or less, to the reduced compounds that are below the photic zone. That includes iron 2 and the thiosulfate. So the capacity gives them an opportunity to access spatially more distant electrons from reduced metals and reduced sulfur compounds. And all of this is contingent on the conductivity of the environment. And because this is a freshwater system, the freshwater itself is not that conductive. And the moment we set these up and we start taking away in our mesocosm experiments the iron oxides, is the moment that this starts to fall apart. So in a freshwater system, it's actually their byproduct of metabolic activity that allows them to increase the conductivity of the matrix in which they live. And that actually allows them to take up electrons from the mineral adjacent to them. This is not an electrical wire. So what you have to realize is all of these minerals, or what we have to realize collectively, is all of these minerals are in a experience, their reduction state is dynamic. And so it's really, I would call it a series of pseudo-oxidations or pseudo-oxidations. And so they may take up an electron from here, and that mineral is in close proximity to another mineral that's more reduced, and so on and so forth. And it's really through electron conduction, or in the case of iron atom exchange, that you end up being able to take up electricity from the environment. And that's more or less what happens. The reason I think this is important is because for too long we've been, as a community, caught up on this idea that our electron donors and acceptors have to come to inside the cell. But for organisms living in an aquatic environment, and in the case of freshwater living in sediments, where you have a lot of minerals that confer a degree of conductivity, or living in the ocean, which is incredibly conductive, I think we need to start thinking more carefully about how extracellular electron transfer matters both to energy metabolism or even extracellular modifications of factors or cofactors that you may be interested in acquiring and so on. And I think it changes the way we think about microbes. We now know that a lot of microbes are actually communicating through electrical impulses. That's been a recent advance as well. So in the case of these iron oxidizers in freshwater photic zones, it now offers a plausible model for how it is they can get past this abiotic competition in a well-lit, well-oxygenated environment, right? So that was the punchline of this work. Is that any questions on this? Everybody cool with that? Okay. So then we started asking now, we know that there's plenty of iron oxidizers in the deep dark sea, and these are different. They're neutrophilic. We don't find them in low pH environments. It's an environment devoid of sunlight. They're not phototrophs. There are a lot of questions about these iron oxidizers in the deep dark ocean, right? We still don't understand exactly how they make a living. Most of them are heterotrophic, right? And that's their sort of source of electrons and the like. But in any case, they too face this challenge of how do you compete with this abiotic reaction. Lately, there's been a lot of attention paid to these microbes in particular, the sort of Zeta proteobacteria. Some of you may have heard about these Zeta proteobacteria. And we got really interested in this question of how do they compete with this abiotic reaction. Now, the iron oxidation rate measurements that people have done to date have been done by taking these cultures that you have grown, putting them in a tube, giving them iron 2, and then you don't wait any more than 5, 10, 15, maybe 20 minutes. You kill the reaction and you look at the ratio of iron 2 to iron 3. And that's how people have made these rate measurements. And we're like, well, that's fine, but that doesn't tell us much about what happens in nature, right? Once this, where they aren't just metabolizing, for 15 minutes. So how does this work in nature? And we just set out to design an experiment where we wanted to make a little mesocosm, right? So it's a serum vial. And we added sources of iron 2 in a solid form. So we either used 0-valent iron, or we used different basalts, and you can imagine any number of iron minerals that we would have tried. And we put them in these mesocosms, along with the bugs and along with some kill controls and the abiotic controls. We sealed them up. They had a little opto dot inside them. Those of you who may not be familiar with this, it's just a little oxygen-sensitive dot. You can read it through the wall of the vessel with a laser and tell how much oxygen is in the headspace. It's really simple. And our students set this up, and really our original sort of notion was that if we do this with living cultures and with dead cultures and abiotic controls, we hypothesized that the living cultures would have the steepest rate of oxygen consumption, and the abiotic controls would have the shallowest rate. It makes sense, right? Because the living cultures, it would be the rate of the bugs plus the rate of the abiotic reaction, and so on and so forth. Everybody cool with that? That's not what happened at all. What happened, and again and again and again, and this is like literally we spent about 7 months making rate measurement after rate measurement after rate measurement, is that treatments with the live iron oxidizers always ended up consuming less oxygen than control. Anybody want to take a guess as to why? Did I write it? Oh yeah, okay. Yeah, this is, oh, I did not write it. What do you guys think this is about? Anybody want to take a guess? How is it that you get less oxygen in your vial with living cells?
Is anyone producing oxygen?
That's a great question. Are they producing oxygen? No. And we have absolutely established that. We did it in the dark, did a whole bunch of other things to inhibit any potential oxygen production. It's not it. Any other guesses? So this bugged us for a long time. And here's what we realized what was happening, is that the iron oxidizers slow down the abiotic consumption rate through the use of exopolysaccharides. So let me go back to show you what's happening here. So essentially, the blue lines up here are our live treatments with PV1. The black lines are our control. You can see the slope is shallower here over 120 hours. Same with tag 1. What happens is, when we introduce the live cells to this 0-valent iron, which is sort of particulate iron, in their media, the abiotic reaction begins to happen. And again, it sort of auto-catalyzes. Very quickly, they produce an exopolysaccharide that not only coats the zero valent iron, but remember those iron oxides I mentioned that are autocatalytic? It aggregates them, which reduces their effective surface to volume ratio and reduces their catalytic efficiency. Does that make sense? So in other words, they're breathing and they're going along just fine, but the reason that we see this decrease in the rate is because they're slowing down the abiotic process. By taking the iron oxides from their own metabolic waste product and sticking them together, which you can actually just physically see with your eyes, right? This is our abiotic control. All of this is rust that's abiotically produced. In the presence of the microbes, you get much larger aggregates. And again, it's a surface to volume ratio issue, because now you've got a ball, and many of the iron oxides in the middle of that ball are not as catalytically active. Sir?
Could some of it be non-stoichiometric mineral phases as well?
Such as what?
Let's say for iron oxides, you could have a whole suite of mineral variations that have just slightly different oxygen.
Yeah. So right now, so we don't have any evidence for that right now, but it is admittedly difficult nut to crack, right? So making those measurements is really, really hard. What we have been doing, though, is working, I don't know if I have these data. No, I don't have it data. But what we have been doing is trying to better understand what are the iron mineral phases that we're seeing, what is the composition of any organic matter in and around the iron oxides and the like. And so what we're primarily seeing is a bunch of ferrohydrite and the like, and no big surprises. The amount of exopolysaccharide that we see associating with the iron is sort of astonishing. It's sort of beyond what you see with typical biofilms. And right now we're working at the Beamline at the synchrotron facility specifically to try and better characterize the organic matrix. But as you probably know, there's lots of evidence for iron associating with organics, right? This isn't necessarily new. What caught our attention is that as we look at the growth of these on zero valent iron and think about what's happening in the deep ocean, wherever we see iron oxides, iron oxidizing bacteria predominantly or as predominant members of the community tends to correspond with slightly lower oxygen regimes. So if you look at where, say, an oxygen minimum zone may intersect the shelf, for example, or just depending on where you are, may intersect the seafloor, just leave it broad, then we start to see extant iron oxidizers that form these mats that have this sort of thick organic matrix covering these sort of iron-rich sediments, right? If the oxygen, dissolved oxygen concentration is too high, or I will call it full ocean saturation, anything, let's call it above 200 micromolar, this does not work. So it works when we see oxygen below about 150 or 140 micromolar, which actually has implications for where you might see iron oxidation in different oceans, right? If you think about the sort of oxygen concentrations in the deep Pacific versus the Atlantic. So that's kind of where we're at with all this. And in particular, a lot of our work has been thinking about what's happening at Loihi and other areas where we see these really thick mats of iron oxidizing bacteria, again, and we see oxygen profiles that have been dissonant with the rate of iron oxidation that people have measured. So I'm sorry, Maya is prattling on.
Do you think that the bacteria are like doing this on purpose to kind of slow down and thus outcompete the abiotic processes, or is it just byproduct EPS production?
That's a great question. So the wonderfully sexy answer would be yes, they're controlling this, but we don't know that that's what's been selected for, right? And because there is the difference between this and the photoferrotroph is they are not in need of light or another factor. So I could argue just as well why not live, I don't know, 6 millimeters below the sediment seawater interface where you might find kind of a nice kind of chemocline there where you have the appropriate oxygen iron concentration. So I don't know. What we do think, or I should back up and tell you that there's certainly evidence for iron oxidizers in many marine sediments, right? But nobody has really sort of systematically gone through and looked at their abundance or their activity and their like. Where we see this kind of phenomenon happening is where we have advective fluid flow from the seafloor, right? Say, again, Loihi or on the fringes of some vents and the like. And there you get these sort of lush mats. Same true with the kind of coastal sediments where you may have the intersection of an O2 min and the like, and maybe you have some degree of tidal pumping. But, sir?
So those are freshwater studies, right?
These are saltwater, sorry.
Saltwater.
Yeah.
So I was going to ask, of course, there is a lot of interesting aspects in the deep sea marine well that's going to have iron-sulfur linkages.
Oh, yeah, right.
Iron-oxygen-sulfur linkages.
Absolutely, yeah.
And so one question is quite geological in context, is the banded iron formation the great oxygenation effect?
Yeah.
And the expansion of these organisms into the deep sea.
Right.
Is there any philosophy that you have on that?
Yeah, so like that's the question that nobody who thinks about iron ever wants to answer. I think, yeah, I am for a while was excited about, go back to my little EET bugs here, as to the possible role they may have in this, because it extends their sphere of influence beyond the immediacy of the cell. So I got to thinking, okay, well, you know, for these sort of, you know, banded formations, you know, is this a sort of more parsimonious explanation for how they might have contributed to it? I'm no longer convinced that's the case. And a lot of it really, I think, has to do with if we take our sort of rates of iron oxidation at their very best, you know, are they sufficient to explain the patterns that we see in the geological record? I'm just not convinced. But that's a little bit above my pay grade. Like, I don't know enough about the banded iron formations to really comment sensibly. With my physiology hat on, though, it strikes me as improbable.
So an inorganic process.
Yeah. Yeah. That seems to be a far more comfortable explanation, knowing what we know about iron oxidizers.
It's got to be widespread.
Yeah, right. So, okay. So essentially, those are the stories I wanted to share with you about iron oxidizers, and raise this question Maya raised is what I wanted to leave you with. Like, this is really cool, and it's advantageous if you are competing with an abiotic process. And I'm excited to consider the possibility that this has been selected for to confer this advantage in a sort of an oxidizing world. And that is not without precedence. Like, we see this among many microbes. Biofilms are a way of controlling your kind of redox regime. So there'd be nothing unusual about that. But I do think it would be wise to consider that there may be other factors that also select for this, that go well beyond. Whatever the case may be, this is definitely advantageous for organisms in the same way that biofilms are advantageous for other organisms. It's advantageous for iron oxidizers. And so a lot of what we're trying to do now is really understand the particulars here. One outcome of this study was in the context of iron-sulfur, right? Iron-sulfur interactions, is we have found a number of biological factors in here that have a surprisingly high affinity for iron that minimizes their reactivity in the presence of sulfur. And that's all, like, literally as it's like 5 days old, right? But this is something I'm working on with Aude Picard, who is now moved on to UNLV, and something we're interested in looking at to better understand how these biologically derived organics are actually influencing the extent to which they might influence iron-sulfur chemistry in some sediments, right? That's kind of where that's at. Okay. And so this is kind of just another way to wrap this up. I think we have to remember that the oxic world can actually be an extreme environment, and we tend to, you know, being aerobes ourselves, tend to think about it in the other way. But you know, life in an aerobic world or in an oxic world is a challenge for anaerobes or organisms that have processes where they're competing with abiotic reactions, including, you know, iron oxidation. And so, to me, I think this is another reminder that sort of geochemistry or geochemical processes and microbial processes are inextricably linked. You know, I continue to learn about geochemistry every day and as a means of helping us better understand what microbes are doing in nature. So, all right, wrapping it up a little bit here, right? Tell you a little bit about whales on the other end of the spectrum. So, as a card-carrying marine biologist, I swore once upon a time I would never study a marine mammal because that's for amateurs, right? Like, screw that. And I'm willing to concede that dolphins still fall in that category for me. It's like, why care about dolphins? I hope I have not pissed anyone off. But this project, the credit really goes to a really bright undergraduate, Annabelle, who came to me and said she was really interested in understanding or studying the microbes that are in the gut of a right whale. She was really interested in looking at their degradation of waxy esters and a few other things. And then I'm like, okay, well, let's start by seeing who's there, and then let's move on from there. And the punchline here is, as we began to look at the microbiome through the lens of the whale's feces, meaning we got on boats and chased whales and collected their poo, we began to realize that there was a really interesting story here. My colleagues Joe Roman and Jim McCarthy have been studying the role of whales and other animals in biological pumping for a while. And what they published on a few years back was that whales, historically at the very least, have been significant nutrient pumps. They feed at depth, they defecate at the surface. Large whales like blue whales, fin whales, and humpbacks, right, they're defecating about 6,000 pounds each day. And in feeding grounds today, for example, where the northern right whale feeds, 75% of the nitrogen used by surface plankton can be pretty clearly tied to whales. Let's be clear here. That's feeding grounds while they're there and they're very active. They are moving a lot of nitrogen to the surface waters, which is pretty cool, right? And so we got interested in thinking about, well, what is the gut microbiome of this right whale in particular? Here's this big feeding ground off of Maine and Massachusetts. Who's in there? Who are the players? What are they doing? What does it all mean? And we started this about the time where Ruth Ley and others were really thinking about microbiomes and how, you know, you are mostly what you eat, and thinking about the role of diet versus phylogeny in your sort of microbiome composition. And the fact of the matter is, the kind of microbes that you have, your sort of microbial diversity, if you will, is influenced by your phylogeny, right, to whom you're related. It's influenced by your diet and other factors, environmental stressors, trauma, other historical events. And we know that phylogenetic history matters, but in some sense, we were really curious to what extent. Why ask this question in the context of whales? Well, because whales are actually a really cool model for asking or studying the role of diet and phylogeny on the microbiome. It's a neat way of asking, what is this sort of phylogenetic like inertia? How much of their ancestry, sort of their ancestry is echoed in their gut microbiome today? Whales are descended from artiodactyl ancestors, and that they are most closely related to hippos, right? And this was really cool because all of these animals, to varying degrees, have some kind of a chambered gut. So hippos do, you know, obviously camels, and we're familiar with cows and the like, and it's because these are herbivores, right? And the chambered gut plays a role in degrading, in supporting a microbial community of anaerobes that are degrading cellulose and other kinds of more challenging carbon that are degraded through, you know, fermenters and the like. And you're like, okay, well, that's fine, until you stop and start thinking about what it means to be a whale versus what it means to, what it means to be a ruminant or an herbivore versus what it means to be an omnivore. And let's take this one step further. Whales they're not even omnivores, they're carnivores. I mean, technically they're not carnivores, but they're meat eaters. Carnivores has its own specific meaning, but colloquially we can call them carnivores. Whales eat other animals. And right whales in particular have a very specific diet. All these baleen whales, though, are carnivores. They're eating shrimps and fishes and the like. They're not filtering out sort of phytoplankton deliberately. That's not what they do. And this was really curious because here's this big giant aquatic mammal that's descended, you know, most closely related to hippos and other herbivores, and it's now a carnivore. So what does a carnivore do with a chambered gut? That's the question that at least I got excited about and got us thinking about how we look at these whales. So this is whale poo. So we working with our colleagues at the New England Aquarium and elsewhere, go around and scoop this up. You may ask, well, what about a contamination from seawater and the like? We can control for that, normalize for that, but I can tell you this is sticky, goopy stuff that definitely sits there as a sticky, goopy mass for a while and is pretty easy to collect with a net. Well, it turns out that to give you some of the highlights here, that their gut microbiome is strikingly distinct from other animals. Whether we're looking at hippos, for example, or toothed whales, and this is data from our paper and other papers, insectivores, carnivores, herbivores, omnivores, and the like. They basically fall out very much on their own. And we started then looking at the gut microbiome through the lens of phylogenetics, which is your typical 16S ribosomal sequencing, through the lens of metagenomics, and through the lens of enzyme assays, looking for activity of particular enzymes. And let me just share with you the punchline. When we look at their I'm going to call them their symbionts, right? Their microbiome bugs. And we ask questions about nitrogen assimilation or protein catabolism. They very much look like other carnivores, right? So if you actually parse the data and you start looking at genes associated with nitrogen metabolism, they fall spot on with lions and tigers and other meat-eating organisms, which is not a surprise, right? These right whales feed on copepods specifically. Like 95% of their diet is this particular kind of copepod. And though they be small, they still, eventually are meaty, I guess, if you get enough of them. So that's what is happening with respect to nitrogen cycling. But what's really striking is that they also host a very kind of conspicuous and prominent group of gut microbes that are chitin degraders, right? And then it like suddenly all made sense. Oh, okay. Because if you're eating copepods, depending on the time of year, you're looking at anywhere from like 12 to 15, maybe 18% of their total carbon is chitin, right? And so in a sense here, you have whales growing microbes in the equivalent of their rumen. They're growing anaerobic fermenters that are breaking down chitin in a manner very similar to what ruminants are doing on land, right? Which are breaking down cellulose. Chitin, like cellulose, is a structural molecule. They're not that different. You can see chitin and cellulose here. And whales, we know, do not have their own chitinase activity. Some animals have an endogenous chitinase. Whales do not. So in the absence of a gut microbiome, they are not accessing the carbon that's tied up in chitin. And so the whale's chambered stomach, which evolved from plant-eating animals, is an ideal, I'm going to call it an exaptation, a word I don't like to use a lot, but you can imagine one of the chambers being dedicated for anaerobic chitin degradation, while other chambers are involved in nitrogen metabolism and the like. And that's our sort of going hypothesis. All of this is based on samples we collected from the poo, and currently we're looking for stranded right whales where we can go out and make collections from their different chambers to better understand the distribution of these bugs inside the whale. And so, that's why we sort of joke about this idea that right whales, are half lion or half camel or half hippo or whatever you want to call it. But I think it's, to me, exciting because it tells us, it's like a window into the evolution of organisms, something that has gone from very likely being an herbivore to now being, to living solely on a meat, living a meat-based life, and how different adaptations can be co-opted or are selected for to solve particular issues. Quickly, I'll tell you a little bit about humpback whales, and then I'll move towards wrapping it up. But I'll just say that we got to thinking about humpback whales in a very similar manner. Humpbacks are cool, right? They have all these different areas that they feed, where there are a lot of fish and plankton and other sort of foodstuffs, and then they have select breeding areas, typically oligotrophic waters that they all congregate at. And what's interesting is whale biologists, of which I'm definitely not one, tell me that when they go to their breeding grounds, they don't eat. And this sort of caught my attention because I was like, well, now wait a minute. You go to a breeding ground to have a baby, and that's when you decide to not eat, to be a little anthropomorphic about it. And so we started doing some sort of simple mass balance, calculations to try and ask, well, if they're migrating, making estimations about their respiration rate, if they're migrating from Alaska to Hawaii, for example, right, how much of their fat stores are gone and how much nitrogen is being catabolized. And really, it's established by the time they get to their winter breeding area, their fat stores are depleted and they're breaking down muscle tissue. What we started to ask is, well, okay, that probably explains why they're not, but they start with ammonia waste, right? All animals do, whether they're, fasting or not. And the punchline here is that ruminants are actually really good at scavenging nitrogen because they eat such a low nitrogen diet. So apparently farmers will often pee on their hay bales before their cows eat it because they have gut microbes that help scavenge urea and recycle it. It's a urea scavenging system that apparently farmers have known about for a while, hence peeing on the hay bales, right? And what we're starting to find just now is echoes of that in the humpback's microbiome. The punchline here is this. They go to their feeding grounds, they get fat and happy, they come here, and now they're in a situation where nitrogen loss is a problem, especially if you're a female making a baby, right? And so we saw one female poo. Like in the history of humpback whale studies, there was actually a colleague who saw it, scooped it up, and it gives us insight into what's happening in the microbiome, and we're starting to see echoes of this nitrogen scavenging that we're trying to put together and better understand. Yes, sir.
With the chitin, is there any way, because the chitin is nitrogen rich, that they can actually store chitin nitrogen?
That's a great question. So first, these are two different whales, but that is a great question, right? And that's one that we started thinking about. In the context of the right whales, it's sort of less relevant because they don't, to my knowledge, exhibit this behavior. And so now that brings us back down to the humpbacks, right? And thinking about what proportion of their diet is chitin, and that's one of the avenues we're literally going down. So I don't have an answer yet. But any other questions about this real quick? All right, and I'm just going to literally, so I've taken up enough of your time, so I'll share this slide with you and wrap it up. But I think one of the, as I mentioned, as a gearhead, we build a lot of widgets, especially to help us do our work in the deep sea. And there are a few things that we've done that have really allowed us to ask some pretty interesting questions. While I was a postdoc at MBARI, I started working on this underwater mass spec. We still use this quite a lot, and it's probably, it's an open design instrument, and thus you can have plans or drawings. There's a full parts list I'll send to you if you want to build one. The instrument itself is about $15,000, and the rest of it is housing, depending on how much you want to spend on a housing. What excites me about this is it's been adapted by a bunch of other labs. So our colleagues at the Max Planck Institute use ours all the time. Our other colleagues at IFRIMER bought 2. JAMSTEC wants to buy a couple, and the University of Washington has 4 that they deploy on the cabled observatories. And I'm excited about this because I feel like we as a community of oceanographers have not been especially effective in sharing our technologies, right? There's so much work to do. And part of that has to do with the kind of the funding culture and other things, but another of it is simply just access and understanding what it takes to make a particular instrument work and so on. And this happened to be one that, honestly, I wish I could tell you that it was full of all sorts of ingenuities that came from my efforts or my effort or the labs, but truly, the fact that this exists is because the semiconductor industry had a demand for smaller mass specs. And at that point, it really became an assembly project with the secret sauce being how do you get gas out of water at 4,000 PSI into a high vacuum, right? So anyhow, that's something if any of you are interested in, I'll tell you more about. We're working on an in situ RNA sampler now for CTDs with McLean Labs. The idea is here is you would take a Niskin bottle off, you would put this on, it has a lanyard, you hook it up, and when you release the lanyard, it goes through a process where it'll filter your fluids, fit on whatever size filter you want, pump in a preservative, seal your sample up, and bring it up. And the idea is to make each of these down and around the $3,000 or $2,000 range, so that you basically replace a Niskin bottle with this widget, and you don't have to build any expensive contraptions. It relies on the infrastructure you have.
You seal the sample, is it at pressure, and you maintain
If necessary, yeah. Yeah, so we have pressure capsules that we can seal with high pressure solenoids, and we have others that we let go to ambient. Yep.
Why specifically for RNA and not DNA?
So I should be more clear. That's, this is our motivation, but you can fix it with anything you want, right? That's compatible with the materials, right? Which is a lot of different things. Underwater blenders, I love telling this story just because it's ridiculous, but when you work in the deep sea at 3,000 meters and you collect an animal and put it on a submarine and bring it to the surface and extract its RNA, what you're looking at is its response to being dragged through 2.5 kilometers of water or whatever, and then put on board a ship. What this allows us to do is lift off the craft, put an animal in there, flood it with our preservative, put the lid back on, it magnetically latches, and blend it. This is the best educational and outreach thing I have ever done. But in the interest of time, I'm going to share this with you and then wrap it up. But microbial fuel cells, they have a great deal of potential in providing electrical power in the deep ocean. Modest amounts, but enough to run things like optodes, transmissometers and fluorometers, magnetometers, acoustic beacons, and the like. And my colleague Claire Reimers and I made the first wireless seafloor network underwater. We went to the Mars Observatory, and then we deployed our microbial fuel cells out to 5 kilometers. And we're able to power our transmissometers and fluorometers and send the data back acoustically to the node. So to me, these kinds of developments that occur worldwide, many of you in this room are doing amazing things. I really want to see us work towards kind of an open access marketplace, right, where we can share our developments so that we can all, sort of further advance our studies. So I'm just going to thank you all for your time, and I want to thank my own lab, Symbionts, and the funding agency. So again, thank you very much. I'll leave you with